Welcome to another episode of the MEGIN Spotlight series. Today we are delighted to have Dimitrios Pantazis as our guest. He's the Principal Research Scientist and Director of the MEG Lab at the McGovern Institute for Brain Research at MIT. Hi Dimitrios, thank you for joining us today.
Hi, thank you for inviting me.
You've been involved in a number of research studies that focus on human visual recognition as well as Alzheimer's disease. Can you please tell our listeners more about your main areas of research interest and why do you use MEG to support this?
So first, let me say that it is a pleasure to participate in the MEGIN Spotlight series. This is a really great initiative to help increase the visibility of MEG research to the general public and also connect researchers from MEG facilities. So we greatly appreciate that. As far as my research goes, I pursued two major lines of research. The first research line involved studying human vision. My interest in this topic was formed when I joined the McGovern Institute to oversee the operation of the MEG core facility at MIT. This is because MIT has a long-standing and continued commitment to vision research. The MIT group of investigators constitute one of the strongest vision groups, employing techniques ranging from genetic manipulations to cellular imaging to non-human private neurophysiology to human fMRI, and now magnetoencephalography, or in short, MEG. MEG stands out as a unique method to non-invasively study the human brain dynamics at a millisecond temporal resolution, which is the time scale that neurons communicate. With MEG, we have an unprecedented opportunity to examine how human visual recognition works. So in my research, I use MEG to resolve human visual representations in time and apply advanced machine learning techniques to identify what information is encoded in the human brain. We also see how the brain constructs static or dynamic representations in response to visual stimuli, enabling us to understand the computational steps of visual processes. My second line of research involves Alzheimer's disease. Given the high temporal resolution, MEG allows us to capture brain oscillatory activity and measure how different brain areas communicate. Using this information, we construct functional brain networks and assess the subtle alterations in brain connectivity that relate to brain disorders and specifically Alzheimer's disease. MEG brain networks are actively pursued by my lab and others as promising and reliable biomarkers for the early diagnosis and monitoring of Alzheimer's disease.
Thank you. So how do you feel MEG helps to connect spatial and temporal brain imaging data? And can you tell us about some of the methods used at MIT to help advance multimodal imaging techniques?
Certainly. Multimodal imaging techniques effectively combine different methods to give us a more complete view of brain function, a view that would not be possible with each method in isolation. Specifically, a comprehensive understanding of the neural basis of brain function requires resolving both the temporal and spatial information of brain activity. MEG measures neuronal activation at a millisecond accuracy, but has a relatively coarse spatial resolution and it does not reveal the precise location of brain signals. On the other hand, the most common type of brain scan, functional MRI or fMRI, identifies the anatomical substrate of neuronal activation, but is too slow to capture brain dynamics. Thus, combining MEG with fMRI has obvious benefits to capitalize on the temporal resolution of one and the spatial resolution of the other method. However, establishing correspondence across imaging modalities remains a challenging problem. Different imaging techniques sample brain activity in fundamentally different ways, relying on different physical principles that necessitate complex transformations from one modality to another. As a result, even though MEG and fMRI offer highly complementary information, the majority of studies are conducted separately by different research groups and a few people combine the two methods. At MIT, to overcome this problem, we developed a new imaging technique called MEG fMRI fusion. This method combines the temporal resolution of MEG with the spatial information of fMRI using a computational approach called representational similarity analysis. Intuitively, the key idea relies on the fact that if two different stimuli, let's say, for example, pictures of faces, evoke similar signals in MEG, they should also evoke similar signals in fMRI. As a result, the comparison of the similarity patterns in response to visual stimuli forms a link between MEG and fMRI signals. Similarity patterns in MEG, informed with millisecond accuracy, can be linked to similarity patterns in fMRI, informed with millimeter accuracy. The combination provides a non-invasive millisecond millimeter accuracy, which would not be possible with either method in isolation. For example, using our novel MEG fMRI fusion method, we produced a first-of-its-kind movie revealing in fine resolution, the activation cascade of the human ventral visual pathway, which is responsible for recognizing the objects we see in front of us.
Thank you. MIT is more unusual as a research-only MEG lab. Why did the McGovern Institute invest in MEG technology and the other areas of research MEG is being involved in?
Indeed, as you said, the MEG facility at MIT is a research-only facility. This means we do not offer clinical service, though we do conduct clinical research together with basic research. This is unlike most MEG facilities that are located in hospitals and conduct both clinical service and research. The addition of a MEG device in our imaging center in 2010 followed the recognition that we need a non-invasive human brain imaging method with high temporal resolution. Since then, the MEG facility has been a valuable resource to our investigators, addressing far-reaching research questions that span low-level visual processing, high-level object scene and face vision, visual computational modeling, attention processing, language processing, neurofeedback, and the study of brain disorders such as Alzheimer's disease, dyslexia, and autism. As you can see, this is an extensive portfolio of research topics reflecting the diverse interests of MIT MEG investigators. These innovative studies have led to prominent research articles in Science, Nature Neuroscience, Nature Communications, and many other top journals in the field. I would also like to say that it is fitting that in MIT we have a state-of-the-art MEG device since the MEG technology was pioneered in MIT. It was pioneered by David Cohen in the early 70s, while he was a member of the MIT's Francis Bitter Magnet Laboratory. There, he conducted the first ever MEG measurements with SQUID sensors. SQUID is short for superconducting quantum interference device, which became the standard type of MEG sensors in the decades that followed. I'm also privileged to say that we have a kind of small museum in our MEG facility featuring Cohen's original notebook with his first ever MEG measurements, an original print of his first MEG article, and one of the first MEG devices dating back to 1970s. So that's a great, I think, a great set for visitors to see.
Absolutely, yeah, absolutely. Let's focus on the Alzheimer's disease research that you do. How do you use data retrieved from MEG technology to support your Alzheimer's disease research?
Alzheimer's disease is what we often call a network-based disease, or in other words, connectopathy. The earliest biomarkers of Alzheimer's disease, amyloid beta plaques and tau tangles, are proteins that accumulate in characteristic spatial patterns that cause specific connectivity disruptions in large-scale brain systems. However, the construction of amyloid beta and tau accumulation maps is only possible with positron emission tomography, which is expensive and involves exposure to radioactivity, making it unsuitable for general population screening. Unlike PET, a potential new biomarker is the disruption of functional brain networks measured by MEG. MEG is non-invasive, low cost, and does not involve radioactivity. It also offers excellent temporal resolution and can therefore capture subtle brain alterations associated with Alzheimer's disease. Given this potential of MEG to capture Alzheimer's-related alterations in brain connectivity, there is a need to develop methods that extract quantitative information from MEG brain networks. My research focuses on developing such tools. Specifically, I develop algorithms called graph embedding methods, which map the high-dimensional MEG brain network data into low-dimensional representations that are easier to analyze and detect Alzheimer's-related features. Using these features, we design biomarkers that can predict when someone will develop mild cognitive impairment, which is the early stage of Alzheimer's disease, or eventually when someone will convert to Alzheimer's disease. We can also identify brain regions with significant MEG network disruptions due to Alzheimer's disease progression. We have found that these regions largely comprise areas in the temporal and frontal cortex and include, for example, the parahippocampal cortex, which has been associated with profound memory deficits in Alzheimer's disease.
Thank you. Thank you very much. How would you describe the future of MEG technology and specifically advances in predicting machine learning and AI in your research field?
Yeah, MEG is already a very powerful neuroimaging method, and work from my lab and others has shown that MEG data capture a wealth of information about the cognitive processing of the human brain. Using this information, we can reveal what a person is seeing, perceiving, or remembering, which one could claim is almost like mind reading, though I don't like really using this term. Using machine learning algorithms called decoding tools, which are often referred to as artificial intelligence in popular science, we can investigate, for example, how the brain encodes complex visual scenes or abstract semantic information. I see a couple of paths that can lead to significant advances moving ahead. One direction is improvements in the analysis of brain signals. This is already happening today through what we call the third revolution in artificial intelligence. Since 2012, we have seen the rapid development of algorithms that now compete with human performance in many tasks. For example, deep neural networks have resulted in computer vision models that recognize objects oftentimes better than humans. I anticipate very soon, in a few years, we will have efficient algorithms that link human vision and computer vision and begin to unlock the secrets of how the human brain solves visual recognition or accomplishes other cognitive tasks. MEG can be instrumental in this effort as it provides valuable human data to compare with computer models. Another direction of significant advances is improvements in MEG hardware. For example, the MEG community is eagerly awaiting a new type of sensors called optically pumped magnetometers, which work in a fundamentally different way than the existing MEG sensors because they do not require cryogenics and can be placed closer to the head. These sensors can lead to higher signal quality and lower cost, radically transforming our research community. More affordable hardware can, in fact, democratize the technology by making it more accessible to a broader community. And with better signal quality, we can capture more information from the brain and at finer spatial scales.
So, as an MEG Lab director and researcher, can you offer any advice to research or medical students about MEG being an interesting career path and how to break into this field? We get this question a lot.
Yeah, this is actually an important question. The MEG community remains relatively small relative to other neuroimaging groups, such as fMRI and EEG groups. And this can often discourage new researchers and medical students from entering the field. However, several recent and ongoing advances will make MEG more powerful and broadly available than ever. I should list a few. For the first time since the early 70s, when the first MEG recordings took place, we have very promising new sensor technologies that can enhance signals and lower the cost of MEG devices. And this can really transform the field, actually. Also, there are new powerful ways to analyze signals, what we call the AI, which can pave the way to new applications and neuroscience advances in the community. There are also software packages and data formats that are increasingly being standardized and becoming more powerful, automating and simplifying the process of MEG signals. And also more and more data sets of MEG, databases of MEG data are becoming publicly available, enabling studies with large populations of both healthy controls and patients, maximizing the potential to detect informative neural signals. And also there are new clinical applications emerging, including traumatic brain injury and dementia, as I mentioned earlier. Considering the above, I would encourage researchers and medical students to enter our field and explore how MEG can answer their research and clinical questions. The best way to break into the field would be to join one or of the many strong research and clinical MEG groups and receive mentorships by MEG experts. Internships and collaborations are great ways to acquire MEG skills. And I would also strongly encourage prospective researchers to strengthen their technical skills, which are important not only for MEG, but practically in most career paths.
Thank you. Dimitrios, thank you so much for making time for us today. This has been a very informative insight into how you are utilizing MEG at the McGovern Institute. Thanks again for your time today.
Thank you. It has been a pleasure.